Good day. This is Dr. T, and welcome to the live stream. So fairly short one probably today. Feel free to ask any questions you'd like into the chat. I think I can see the chat. But yeah, so welcome. And yeah, I'm going to get started. So today, the kind of topic du jour. No questions in the discussion forums are on the post. But the question, kind of topic du jour is going to be, hypothesis, and what is it? So, grading tests, and the first set of tests, usually in the fall, ends up having a question, something about the scientific method, or how science is done, or one of those things. And for this one, I got a lot of responses about the scientific method that included things like a hypothesis is an educated guess, and not quite. No, they're a somewhat different category. So let's kind of think back a little bit more on scientific method, because truth be told, what we learn in grade school or middle school about this is the scientific method. No, that's not the scientific method. That's not how we do this. Instead, typically what's really happening is, as a scientist, I'm going in there, and there's a couple of scenarios. One, I could be doing what Kuhn would have called normal science. So in that case, I've got my understanding of how the world works. I'll call this the model. This is kind of what Kuhn would have called it, what a few other folks would have done. And Kuhn was a 1940s, 50s, 60s era physicist that wrote influential work on the philosophy of science and how science is really done. And so, his thought was, okay, it's like you got all, how we understand this one part of reality, whatever that might happen to be. And there's holes in that. There's things that we can deduce. There's things that we can logically figure out, but we don't know if they're true. And so those logical deductions, those predictions based on our understanding, that's our hypothesis. Okay, so I think this, and this are true. And if this all is true, then I could do an experiment. And the results of this experiment, assuming what I understand to be correct is correct, would do this. Now, that is a lot of what we do, but that's not everything. Quite often, we don't have enough of a model. We need model building experiments. We need hypothesis generating ones. We need to know what on earth we are doing before we can ask a good question. So one, hypothesis isn't an educated guess. It is basically a prediction of, if I know what on earth is going on, this is the way the world works. So it is kind of the confirmation. It is the prediction for an experiment. It's the thing that I'm going to match up. Now, once again, the scientific method, quite often we're not going through this whole hypothesis approach because we can't. We need to know more. So a classic case would be my undergraduate research. In this, I was studying an enzyme called VATPase, and this is used to acidify parts of a cell. Basically, it's pumping protons from one end of a membrane and shoving them to the other, uses ATP as power. It's basically the inverse of the ATP synthase that's in the electronic transport chain. And so we're looking at it's gigantic protein. We're looking at a small part of it. And so what we're going to start out with, and what we started out with, by we, I mean my advisor, this was undergraduate research. I was doing a tiny little part of a very big question. And so what she had done was she did a sequence alignment. And so this one goes by, you take the genomic sequence of a bunch of different organisms, you convert that into the amino acid sequence for that protein. And quite often you just start with the amino acid sequence. You can look it up in a database. And you got different organisms that are not all that closely related. So you might have human and E. coli and rice and mouse and Arabidopsis, and you're all over the place. But if you've got the same enzyme and it does the same thing, then the idea here is certain amino acids will be important. They're part of what's going on. And those have to be what's called conserved. They have to be the same. If they're not the same, then it won't work. It won't do the job. But other amino acids won't be conserved. They can be swapped out for another amino acid. You could add more, cut them out altogether. And those amino acids aren't all that important for the function of the enzyme. So what we can do is a sequence alignment. We can take a computer algorithm, and anymore it's not a particularly powerful software that's needed to do this. Frankly, a tablet I'm quite certain could run it if you made the app. But you take the software, you plug in your amino acid sequences, and it does its best to kind of try and line them up. And then that will tell you which amino acids are conserved, which are semi-conserved, which means they're swapped out, but they're swapped out for amino acids that are pretty close to the same amino acid that they were originally. And then ones that are just, it was whatever. They may get cut out, they may get swapped around, they're not conserved. And so now what I do, or what my PI did done, was she had looked at, okay, these amino acids are conserved. Maybe they're important. So now we've got kind of this like partial hypothesis, but it's not really a full-on fledged hypothesis. What we really want to do is this a protein or this enzyme functions by doing this, this, and this. That's what we want our hypothesis to be, but we haven't gotten there yet. So now, we could say we had a hypothesis that this was important, and you can argue that, but truth be told, that's back arguing. That's taking the information and kind of feeding it backwards, because we had planned on going before. These are not separate experiments. These are just parts of the steps. So now, okay, we've got these. Let's change them. Let's do what's called site-directed immunogenesis. We're going to modify this, genetically modify our organism. In this case was yeast, because yeast is easy to work with. And we will force that amino acid to be something different. If the enzyme keeps working, well, it wasn't that important. If it doesn't keep working, then it must have been important. If it was sort of working, it must have been kind of important, And so we modified the amino acids. We saw if the enzyme kept working. We then gathered more data by figuring out a structure of the amino acid. We didn't do that. That had been done by other folks. And then by knowing the structure of the protein as a whole, what amino acids were important by site-directed immunogenesis and by sequence alignment, then we could start really developing a model of how that worked. And now that we got the model, we can actually start getting a little bit more interesting hypotheses to start understanding it. So you can force a hypothesis into each one of these steps, because this is kind of the natural way. If you have, here's my experiment, if-then scenarios. But in some respects, when we're doing the sequence alignment especially, I'm just going to see what I get. I don't hypothesize that this particular amino acid is going to be conserved. I just know that the conserved ones are probably going to make a nice hypothesis for later on. So I'm just going to throw this in. I'm going to generate a hypothesis from this one. Now, once you get into the site-directed immunogenesis, then you can really argue that, yeah, this has been conserved, therefore I hypothesize it's important. And that becomes much more of a legitimate hypothesis. But once you're going to sequence alignment, at that first step, it's like something's got to be important, but I don't know. So I'm generating enough information to build enough of a model that I can start generating and making a hypothesis. So scientific method does not necessarily end up being research, hypothesize, experiment, conclude, or whatever you want. That is part of it. That is definitely done. That's kind of the gold standard. That's what we want to do. But there's a lot more of just, I'm going to throw darts until I hit something enough interesting that I can actually make a hypothesis, which does happen. It's frowned upon. It's not really frowned upon as much as like, oh, great, I got to do this. It's not as desired, but it definitely is a major part. And then, of course, there's the whole write-up, etc., which we always leave out. We don't usually tell students how much writing is in the sciences. Quite often you spend as much time out of lab writing up reports as you do in lab actually doing experiments, if not more. Which is one of those is like, oh, great. No one told me this until it's kind of too late to change my major kind of thing. Things turned out well, though. But yeah, that's kind of the topic for today. Here in Georgia, we've got the remnants of a hurricane coming. Not the whole thing, thankfully. Sorry about that. But yeah, so I hope things are going well. That was kind of my topic. Once again, Q&A live stream with not too many questions. Going to be kind of on the short side. That said, we have tests coming. Actually, not really. I don't think too many of my classes have tests coming up anytime soon. So here in a few weeks, there'll be a few more questions. But until then, if you've got questions, feel free to chuck them in the comment section of a video. Or if you're one of my students, chuck them on any of the Q&A discussion boards for each class. So with that said, have a wonderful day. If you're in the path of the storm, thoughts and prayers for you. I hope everything goes well. And I'll see you next time.